All right, so very good morning to all of you and welcome to APA India 2021. This is our webinar number 4 and today's session is on regulated bioanalysis. We still have close to around 6 to 7 minutes to go for. So what we will do is we will quickly go for a promotional video, a small video from Phenomenix. So once again, welcome to all the participants. We will start within the next 7 to 8 minutes. And meanwhile, let us go through this small video from Phenomenix. So today is 10th July, webinar number 4, regulated bioanalysis. And the monthly theme, as last month, in the month of June, we talked about repurposed drugs, vaccines, and diagnostic development to combat emergency disease outbreaks. For the month of July, the session, the monthly theme is technological advancement. So all the talks, be it regulated bioanalysis, discovery, biotransformation, or CMC, we will focus around technological advancements. So today, we are going to have a small video from Phenomenix. So Phenomenix is basically a global technology leader committed to developing novel analytical chemistry solutions that solve the separation and purification challenges of researchers in industrial, clinical, government, and academic laboratories. From drug discovery and pharmaceutical development to food safety and environmental analysis, Phenomenix chromatography products, technical services, and chemical reference standards accelerate science and help researchers improve global health and well-being. Selected by the Wall Street Journal as an exceptional workplace, Phenomenix is defined by its passionate people, dynamic culture, and distinctive ingenuity. These common characteristics unify the Phenomenix companies worldwide behind their mission to promote the growth, prosperity, and well-being of those we serve, our customers, employees, and humanity. So that's one of our really esteemed sponsor, Phenomenix. And I would like to mention that since the inception of APA India chapter, Phenomenix has always been supportive of this meeting, and they have always participated, actively supported APA India chapters since the inception back in 2009. So with this, let us go to a small video from Phenomenix.
Kim. My role here is Senior Director of Global Marketing. I always tell people that the drugs that you put in your body, the food that you put in, that you ingest, these things are all things that ultimately run through a product that we manufacture. We're one of those behind-the-scenes players that no one knows of, but life doesn't really function well without providers like Phenomenix in the market.
My name is Sean Orlowitz. I currently work in business development for food, environmental, and cannabis. We're trying to understand what our clients do with our columns and then using our expertise in chromatography to improve that.
My name is Srinivasa Rao. I'm a senior scientist at Phenomenix. Once the product is made, we do extensive customer testing. It will start right when we are in the prototype stage, and then it will go all the way until we release the product, and we continue to serve the customers as they are our highest priority.
My name is Erin Tappan. I am the Senior Manager Director of the Global Business Unit Marketing Group. My team is responsible for staying in tune with the various industries that we serve, so working together with customers, forming those relationships, understanding what it is that they need, what they're doing, where is the industry going.
My name is Manal Salim. I work at Phenomenex as a product manufacturing quality control manager. We do come together as a team to develop a product to make sure it's being made for what it's meant to be doing.
My name is Rupali Ray. I'm the Human Resources Manager here at Phenomenex. You can't be in Phenomenex and not feel good about what you do. When we talk to the R&D folks and we talk to the sales folks as to where a product finally landed and who it helped. You know, whatever good we do, it really makes you feel good at the end of the day to go to bed at night knowing that you are contributing. You are doing your bit.
My name is Ike Emme. I've been with Phenomenex for 30 years. I work here as a manufacturing tech, and that 30 years seems to have gone by so fast. In Phenomenex, we all work as a team. It doesn't matter if you're the top notch or whatever, because you never know where the idea will come from. It could be from the rock bottom, it could be from the top.
My name's Terrell Matthews. I'm the VP of Sales at a company like Phenomenex. When you have retention of employees like we have, you make a difference. You feel as though you're making an impact. We've become part of a bigger company now. Phenomenex now is part of Danaher. But without a doubt, the reason why we have people here for 30 years, the day that the founder opened the doors, that person is still here, is because he's making an impact every day.
My name is Tim Edgefield. I am the shipping lead in the shipping department. I think people take a lot of pride in what they do, although it looks like a lot of bins and hardware on the shelves. At the end of the day, the finished product comes from a lot of hands touching it and a lot of people with pride and dedication.
One of the reasons why I like what I do is we are contributing towards the goodness of humankind. We're not just here to make money. We're here to help the science and be better for our humanity overall.
Whatever we can do to pull together and come up with some type of solution where we can help people identify faster, quicker, better, that's a passion for people here. It actually fuels that family environment because we're all working together towards one common problem that we know we're making a difference.
We have a real investment and commitment here by the people, and I think that's reflected in the way that we approach our markets and the way that we service our customers.
We want to be here because we believe in our products. We believe in the quality. We believe that we're giving our customers a better product to work with, something that's going to provide a better answer, a faster answer. The pace of innovation, the things that we're trying to do, the direction that the company is going, you love it. You're swimming with your friends and you're having a good time and you're trying to impact the world a little bit positively every day.
In our world, we were 1,000 people. Now, we're over 69,000 associates around the world that we can learn from each other's experiences, and not make the same mistakes that I would have made it if we were by ourselves. It's a great combination between Phenomenex and Danaher World together.
All right, so a very good morning to all of you, and welcome to APA India 2021. We were just going through the Phenomenex promotional video of one of our sponsors. So let me, all the attendees who have just joined, let me welcome you all to AP India 2021, webinar number 4. Today's session is regulated bioanalysis, and we are going to focus this entire month on technological advancements. So very quickly, a special thanks to our organizers, the presiding chairs, chair-elect of all the sessions, including regulated bioanalysis, discovery, biotransformation, and chemistry, manufacturing, and control. So a special thanks to the chair and chair-elects, Anurag, Ravi, Amarnath, Shefali, Ravi, Kumar Trivedi, Jignesh, Prakash, Samula, Rajesh, and Purushottam. Special thanks to all the very active team, which is, for each of the sessions, they are supporting the chair and the chair-elects, regulated bioanalysis, Darshana, Srinivas, Patanjali, Venu, Peter, and Mallikarjun Dikshit. Same way, special thanks to Discovery Bioanalysis, Deepak, Jonathan Josephs, Prashant, Vijay, Bhavesh, Ravi Shankar, and Shrikant. At Biotransformation, Rama, Kaushik, Selvan, Ujjwal, Mohammed Jainuddin, Zakir, and Vinod. And for chemistry, manufacturing, and control, special thanks to all the organizing team, including Sailesh, Hugh, Satish, Anurag, Pankaj, Hemant, Ajit, and Vishal. Also, as you all know, without the support from sponsors, it's not possible to do an event of this level. And so we are so very grateful to all our sponsors who have helped us throughout since the inception of AP India chapter since 2009. So special thanks to all our sponsors, Shimadzu, SciEx, Zevsci, Phenomenix, Thermo Fisher, Agilent, Syngene, USP, Netco Pharma, Vida Clinical Research, Bristol Myers Squibb, Zoetis, In Vitro Med Labs, Origin, and Nanotemper. We are also thankful to the collaborators. This year we have collaborated with Life Sciences Sector Skill Development Council, which comes under Ministry of Skill Development and Entrepreneurship. So special thanks to our collaborator, Life Science Sector Skill Development Council as well. Couple of announcements before I invite the session chair about the poster session. Would like to request all the participants who are attending today's webinar, especially the students and researchers, that please submit your poster abstract. The last date is 1st of August. All we request for is 200 to 400 words of poster abstract, and it should be within the areas of drug discovery, biotransformation, regulated bioanalysis, or chemistry, manufacturing, and control. So please share before 1st of August, and then we will go for the first round of screening in the month of September, and the final poster session would be held on 2nd October, where the poster presenter will get 6 to 8 minutes to present their poster, the shortlisted ones. And then finally, the committee will select total 9 winners, 3 best outstanding posters for first prize, 3 best outstanding poster for second prize, and 3 best outstanding posters for third prize. Again, for just to keep you all engaged and for the excitement and thrill for the attendees, we always do in in-person meeting, we do a raffle draw. Since this year we are doing an online meeting, we decided to do a small online draw, but this would be done at the end of the webinar. We'll be running one webinar poll. So we have already done it twice last month, and then there were a couple of people who wanted to confirm that how do we ensure how do they ensure that their name is there in the list. So just wanted to clarify that towards the end of the webinar, when both the talks of all the 3 talks are over, we actually run a webinar poll where there are a few basic questions about the webinar. You have to participate in that poll. You have to select some answers. And then once you submit that poll, we get a automated report, all those who participated in that poll. So I get a list of that in an Excel document, which I'll display today. So you will see, so all those attendees who are participating in the poll, their names would be included in the lucky draw. So far, 6 winners are there, like Matru Prasad, Vasudha, Siddharth, Vinay, Shrim Sharma, and Deepa Nagesh. So all of you would have already received an email. Just follow the instructions on that, and you will have your surprise gift with you. So finally, For Q&A, while the talk is going on, as you know, as an attendee, you can see and you can listen to the talk. But if you have any questions, technical queries, there is a tab below the screen called as Q&A. Please feel free to post your questions anytime during the talk, so that at the end of the talk, moderator can take up those questions for the speaker. Still, if you have any further questions and if you are not able to take up all the questions during the webinar, feel free to post your queries, questions, technical questions to bostondb21@gmail.com, and we will go back to each of the speakers and try and get your answers for you. Again, if you want to participate in the poll, then that will happen at the end of the webinar, after both the talks, after the panel discussion. And finally, the webinar talks, right now it is live streamed on the YouTube, but later on also, we are going to upload this talk, an entire webinar on a YouTube channel of Boston International. So you can subscribe that. And of course, it will be done only after we have a permission, a written permission from the consent from the speaker. So with that, I think we are just on time. Let us get started, and let me invite our session chair for today, Dr. Shefali Gupta, and for her inaugural address, and then take up the further proceedings. So over to you, Shefali.
Yeah. Thank you, Deepak. Welcome all. Good morning on this bright, sunny morning today. I invite all of you to the technological advances in regulated bioanalysis. Let me introduce myself first. I'm Dr. Shefali Gupta, a pathologist with over 20 years of experience. Initial 10 years were spent in the clinical laboratory. And after that, in the last 10 years, I have been in the regulatory environment with Cliantha Research Limited, serving as a senior director to regulated large molecule bioanalysis, wherein we do PK, immunogenicity, and cell-based analysis, and a lot of biomarker testing. So over the last few years, the industry has seen many technologies maturing. You know, we've seen cell-based assays coming in, genetics, and so many of hybrid technologies coming in. And as they become available, the technology scientist has observed that implementing these technologies in the regulated environment has become increasingly challenging. For one, the scientific developments and regulatory expectations often may not go hand in hand, and they often are because of the emergent technologies, are not completely outlined as yet. At the same time, the pharmaceutical industry has become increasingly risk-averse in their response to these real and perceived higher expectations in regulated bioanalysis. As a consequence of this, the potential result of over-interpretation of the guidance or occasional widespread or premature implementation of the responses to the health authority inspections, industry has been contributing significantly to raising the bar on these processes. Also, the community, has not been being satisfied with the current tools. There is some level of potential complacency, if we could say so, on in the regulated bioanalytical community because of the existing technologies such as LCMS, and LBAs, which are serving so well to us. Hence, the question that was there in the early 1990s, what's next after LCMS, or after RIA, GCs, or LCUVs, is not certainly in the context, which was a key question in the 1990s, is also the question that we are facing today to validate these new tools. And therefore, we so today we have a discussion to facilitate the evaluation and subsequent implementation of these innovative technologies. For this, I would like to invite Jesse from Somru Biosciences, which is a case study of method validation. She's speaking today on method validation using integrated bioanalytical method validation software for pharmacokinetic and immunogenicity assays. Just a brief introduction to her. Just give me one second. Yeah. So Jesse Rix is a bioanalytical scientist at Somru Biosciences, Prince Edward Island, Canada. Her role is to serve as a scientific and technical lead for biopharmaceutical companies. She plays a role in validation and applications of bioanalytical assays to comply with scientific and regulatory requirements. Jessie has 7+ years of professional research experiences of regulated bioanalysis in developing, validating, and applying bioanalytical methods to support clinical studies of biosimilar products. In the laboratory, she has an experience on developing and transferring bioanalytical methods for the detection and characterization of large molecules, endogenous molecules, hormones, and viral particles. Other lab experiences include tissue culturing, monoclonal antibody generation, immuno-PCR, and immunogenicity assessment. Jessie also has an experience in testing and validation of bioanalytical software and is a certified training manager for the bioanalytical and laboratory productivity suite of Aegis software developed by Somru Biosciences, Inc. Jessie has published in several research articles in peer-reviewed scientific journals, international journals like Bioanalysis, Environmental Technology and Innovation, and Canadian Aquaculture, R&D Review 2019, etc. So,
सेम ही है सर, आपने ऑलरेडी ले लिए एग्रीज का.
I invite Jessie to please start with her talk.
Case study है कुछ.
Thank you, Shefali. And while Jessie is going to share her screen, would like to humbly request to all the panelists to please kindly mute your lines. You can only when you have to speak. Thank you. Over to you, Jessie. Thank you, Shefali.
Thank you, Deepa.
Thank you for that wonderful introduction. I'm going to share my screen. Just give me one moment. While I figure that out again. Sorry, it's having an issue where I am unable to share my presentation correctly. Just one moment. All right. Sorry, having an issue. It's having that issue where I can't do it in the presenter mode like previously, and I don't know how I corrected it previously.
No worries.
So,
I mean, you can share the screen and maybe we can look at, you have to select that particular window.
Okay, let me just try this 1 second. I'm going to do this. All right. Okay, it's fine. I just won't use my notes, so I might get a little off topic. So I will share from here. So, good morning. Today I'm going to go over the Agris suite of software platform overview. So the discussion today will be the Agris platform overview, the pharmacokinetic validation module, the immunogenicity validation module, and a brief discussion of the productivity module and a small Q&A. So what is the Agris platform? So Agris is a comprehensive software platform for a bioanalytical laboratory. It consists of two main modules, bioanalytical and productivity. Each module has multiple components. The bioanalytical module has 6 components: pharmacokinetic, immunogenicity, biomarker, biosimilar, trending, and sample analysis. The productivity module contains 3 components: inventory, eWorksheet, also known as eLabX, and RFID sample tracking. Each component is designed to work independently as well as in tandem. So it is good for clients who may just be looking for 1 or 2 of these components for validation or sample analysis or other components. And it also can work in tandem together. So multiple components will all work together well. So the Agris is the 21 CFR Part 11 compliant, which is an important FDA regulatory requirements. And it is very intuitive for the interface to be used on a wizard-based workflow. There is an optional direct link to an LMIS or data system, and Somero Bioscience can either offer validation scripts or customize scripts to the client's needs. So like I mentioned previously, the bioanalytical modules, there are 6 that work together. Again, they can also work independently or in pairs. So we have our ADA, our immunogenicity, biosimilar, trending, pharmacokinetic, biomarker, and sample analysis. The productivity modules, the 3 components are built, comprised of IntelliLab, eLabX, and sample tracking. So IntelliLab module actually then contains multiple modules within it. So companies can go from have just an inventory system, or they can track everything from the beginning to the end. Okay, I actually just figured something out. So I'm going to share this correctly now. And is that screen being shared?
Yes, I mean, you're good to go.
So can you don't see all my notes now?
No, we don't see that, yeah.
Okay, good. So IntelliLab, let me move this. So IntelliLab productivity module, including eLabX, is designed to streamline day-to-day laboratory activities involving inventory management and electronic laboratory notebook. The IntelliLab inventory management system is designed to manage the life cycle of laboratory equipment, reagents, and consumables. Within the IntelliLab system, users are able to manage ordering, reagent use, and location traceability, as well as equipment logs and maintenance. The eLabX system is designed to integrate the laboratory digitally at the bench level, as this user-friendly tool enables analysts to structure and manage their workload by creating technique-specific worksheets, entering assay execution details in real time, as well as allowing for the quality control and quality assurance review. The main functionalities of eLabX includes eWorksheet design, eWorksheet execution, and eWorksheet review. Agris IntelliLab provides an audit trail detailing changes made to the inventory and records, the eWorksheet design, execution, and review. The Agris, The Agris bioanalytical system has been designed to handle complex clinical study protocols for both the pharmacokinetic and immunogenicity studies. Clinical, preclinical, and discovery study protocols, including crossover, parallel, multiple dose, and TKPK non-clinical studies are supported. When a study is created, the default system configuration values will be applied to the study to achieve consistency while maintaining maximum flexibility. The study configuration data may be updated as needed, either manually or via importing an existing study configuration from a previous study. Agris provides an audit trail detailing any and all changes made at the program project study level. Modifications to the study configurations are tracked for each.
Jesse, sorry to interrupt you. Just wanted to reconfirm, you are on a slide of productivity modules, right? Where we see these three triangles.
Yes.
Yeah, okay, go ahead, please.
Yep, sorry. So the RFID sample tracking, sample management software designed to be easy to use while providing powerful lab sample tracking and workflow features, from managing sample registration and freezer storage to guiding sample processing tasks and data capture within workflows. Sample Manager tracks the entire life cycles of samples within your lab. So the Agris Cloud Solution. So Agris Cloud Solution. Security, the data is always encrypted in transit to and from the cloud, as well as when it is in rest in the system. A well-defined role-based policy designated people only have access to the subsystems. Data storage in the cloud is scalable, automatically backed up daily, well-designed business continuity, and disaster recovery plan, benefit of data redundancy. Collaboration effectively allows scientists to work from anywhere at the heightened connectivity, improves efficiency and productivity. I can very much attest to this, as the last 6 months I've been working from Prince Edward Island, Canada, with the team in Vita in Ingenuity Biosciences. Daily, we are in communication via video and in the Agris software. So the other cloud solutions are the total cost of ownership. So Agris is a very cost-saving system, as it is, it reduces there's no hardware to acquire and maintain, and the total cost is minimized. Tech support is near real time, and maintenance is included in our service fees. It has no maintenance and near unlimited scalability, and is highly reliable. The Agris Data Analyzer is the statistical analysis engine. It uses the standard R library package. The R packages are extensions to the R statistical programming language. The R packages contain code, data, and documentation in a standardized collection. It has been validated by multiple well-recognized organizations in both pharma, financial institutions, etc. And it communicates with the Agris base via ESB using HTTP API. So the Agris has a report function as well. There's a powerful reporting server. The reports can be generated in multiple different formats, in Doc, Excel, PDF, standard report template, or user-defined template. So Samru can provide our standard reporting template, but it is also extremely customizable based on the client's needs. And the baseline for all of the Samru templates that are available are the minimum FDA regulations for bioanalysis. It provides standard HTTP API for communication and has custom integration options with third-party document management systems. So it pulls information directly from the study and is highly configurable. So some key components in the 21 CFR Part 11, the FDA requirement for using software in a regulated environment. It ensures that the software is compliant with the FDA and the audit trail electronic signature and single user login. So some of these key features are managing user roles, logins and roles for the application, audit trail and electronic signature is saved to the file, the workflow, user management audit trails, and tamper evidence file structure. And archiving, the audit trail is automated. It is secure and cannot be edited. It is traceable and archivable. The audit trail includes identification of user, date and timestamp, record ID, original value, new value, reason for change, and the electronic signature. So the archiving, the data is locked in stores in archive directory with limited access. There will be an archivist, and to obtain any of the information, once it has been archived, there will be required to be an archive request, an approval from the archivist, and a read-only access with a limited time frame. So on to the bioanalytical validation modules, the immunogenicity and the pharmacokinetic. The agarose pharmacokinetic module we'll discuss first. In the agarose pharmacokinetic module, anyone who's familiar with, you will see here in our study menu is broken down with the regular and FDA required steps. So we have our configuration, our validation plan, calibration curve, accuracy and precision, including inter and intra, selectivity, dilutional linearity and hook effect, sensitivity, specificity, stability, and then a validation report. So agarose is going to complete all of the data analysis and the planning and reporting of the analysis. So here we can see the calibration curve that has been generated by agarose. So agarose allows the ability to choose the regression type, the tuning parameters, and the weighting for these calibration curves. And this again is all configurable and is dependent on the configuration set at the prior to the validation plan being signed. And so previously was our graph, and then these are our results that are being shared. So we have our ID, our calibrator type, concentration, raw data, mean data, our percent CV, our calculated concentration, and our mean concentration with a percent bias. Once the calibration curve has been analyzed, we will do, the agarose does the control evaluation, sharing the same stats here for each of our controls. So as a regular PK analysis, we will have an HPC, a MPC, and an LPC, and our evaluation here. So it isn't necessarily showing here, but if any of our bias or CVs are out of spec, then these will be highlighted in red. So for the configuration, if our percent CV should be less than 20 and this was greater than 20, it would be flagged in red and it would be presented that way. So some summary tables for accuracy and precision. Here you can see our total error calculation, and each of these tabs in the software you will be able to click through and see each of the statistics that have been calculated by agarose. And the accuracy and precision statistics here, the precision table, summary table here. So this is an example of our selectivity result. Here we will have each of the serum types. In this example, they are both normal, but it does have the capability to include diseased or lipid or any other serum types. And then in each of the serum types, you will have your expected concentration, the and the actual concentration. In the report, it is included with the percent bias and other calculations as well. So for the immunogenicity component, we have the study menu again for each of the calculations and assays that are going to take place. So again, we have the configuration for all of the statistics that are going to take place. There will be a validation plan document and a template, that's the word I'm looking for, and a template. Once the validation plan has been signed off, you can go ahead and start with each of your assays. So we have our screening point, cut point, confirmatory cut point, titer cut point, and titer precision. Precision, inter, and intra, again, including robustness, selectivity, screening and confirmatory, drug tolerance, sensitivity, hook effect, stability, and again, all of your data would be then compiled in a final report. So the screening cut point is one of the major calculations that takes a lot of time and calculations. So the calculation flow here for Agris is based on the most recent FDA guidance and industry best practices. So there's no need for manual formatting, additional statisticians. Agris is performing the statistical analysis and then will provide recommendations for the flow based on results. So you can see you will do an NC evaluation. You can choose your options of mean, median of your NC evaluation. Possible to normalize data, remove outliers, analytical outliers, and also remove biological outliers. The Agris will do its calculations, and then if there is skewedness or issues, you can do different approaches, either the parametric approach or the non-parametric approach. And again, Agris is going to give you the best options and will give the recommendations based on your actual data. So for the confirmatory cut point, slightly different from the screening cut point, Agris is going to calculate the percent inhibition, identify the analytical and biological outliers. It's going to exclude the outliers and evaluate the distribution. So once this is done, again, Agris will give you the recommendation for the parametric method or to transform different methods of transforming, log transformation, etc. And then again, it will give you those recommendations and you can finally get your cut point. So an example of the screening cut point workflow is here. Some of our results. So Agris for this data has given us the recommendation for a floating cut point. And there's some very tiny writing on this slide, but it's going to move forward in the correct methods and then give you your cut point calculation based on your data sets, and your final cut point will be calculated. So in the immunogenicity module, I've given just a few more examples of the calculations here. Some of the drug tolerance results, again, so we've run all of our assays and incorporated all of our data into Agris, and now Agris is doing our calculations and going to give a drug tolerance for our ADA. So here you can see the ADA concentration. So we've used a concentration of 500, I believe, micrograms or nanograms here. And then this is the actual drug concentration. So we've made a dilution curve of our drug, and Agris has done these the calculations and given us a drug tolerance for this assay at 25 micrograms per mil of this drug at 500 nanograms of our ADA. So in the immunogenicity module, we can also evaluate our selectivity. Here is some of our selectivity results for different IDs, and we will have our selectivity for various concentrations, our mean, and our positive or negative results for these. And then a actual selectivity pass or fail mark for the assay itself. So for the immunogenicity, we also have a summary precision available for all of the runs. So the summary precision table will look like this. We will have raw data value criteria, signal to noise criteria, and inhibited criteria. We will have different data types, our negative, LPC, and HPC. And you can see our means, our medians, our standard deviation, CV, etc. Upper limit, lower limit, and you can add comments in here as well. And again, all of these data tables are available to be generated into the report as well. So for the validation plan and report, the validation plan again, is configurable and customizable dependent on the client's needs. It contains 3 types of data: free-form text, which allows the analysts or writers to edit the report, add any information that is required. We have non-editable template information. So there should parts of the report that should not be edited, such as the compliance statement, signature lines, etc. And then all of the data from Agris in the plan would be the configuration plan. So once that's signed off, then you can initiate your study. And then once the study is completed, we can move on to our validation report. The report, again, is configurable and customizable. Agris generates the data tables, which are then inserted into the report. So the free-form text, again, you can add comments, edits, etc. There will be non-editable template information, your compliance statement, etc. And then data from the Agris analysis. So all of your data tables will just be plug and play. You will click on a radio here and a radio there, and all of your data will be inserted into your report. Once your report has been completed, reviewed, finalized, it will be transformed into a Word document or a PDF, and all of that will be then in your report. So just a very brief summary of some of the productivity modules. I gave a brief overview at the beginning, but some of the productivity modules are IntelliLab, our eLabX, and our sample tracking. So for the inventory module, there is real-time inventory asset tracking. So from the moment that you request an item to be ordered, it can be approved by a manager or a senior leader, ordered from purchasing. Once it is received, it is immediately put into the inventory system. Real-time tracking of checking in and out of items, and based on ID or barcode. So you can submit your requests, approve requests, receive inventory, and critical reagent lifecycle management. All of this is then downloadable in a report. So then eLabX, our electronic assay worksheet. So once a assay has been developed and an SOP has been signed off and approved, you will make an e-worksheet based on that. It has built-in QC and QA functionalities based on automated flagging of inconsistent data and has actually been developed and released. So then the sample tracking module, real-time sample tracking to minimize error, and is also developed. So that is it for my slides. I can open it up to Q&A. Also, if there are very tech questions related to computer programming, etc., we have an email here that you can direct those questions to.
Thank you, Jesse. May I now request the moderator, Shefali, Amarnath, and Srinivas to take up the questions, and would request Jesse to, if you can stop the sharing.
Yep. Yeah, thanks, Deepak. Srinivas, would you like to please ask your question?
Yeah, sure. Thank Jesse.
Thank you, Jesse. It was a wonderful talk and it was really impressive. The software really looks extremely impressive and a very complete one, you know, including the PK and the immunogenicity validation and the study sample analysis requirements with a real-time sample tracking of the sample tracking happening along with statistical analytical tours, which reduces the the entire work back and forth for the lab so much in between departments. So I think it'll really be of big help for the large molecule bioanalysis analysts. And thank you for sharing everything. Let's yeah. Please, Srinivas.
Thanks, Jesse. Just quickly wanted to understand where does this software sit? Like, and what is the data source? Is it the application software or is it the laboratory information management system?
So it is a cloud-based software that we are deploying and taking care of remotely. So the R suite of statistics have been validated, and here, just one moment, I'm going to open something. So, sorry, I'm not sure I fully understand your question. Can you expand upon that?
Srini, your voice is not coming.
Sorry, I was asking, what is the source of data for your software? Is it coming from the application software or from the laboratory information management system?
It is coming from the sorry, I'm trying to read this. The laboratory management system. So it is the R library package. So the R packages are the extension and the statistical programming language that has been validated from multiple recognized organizations. Is that answer?
No, it's more to understand the kind of handshaking with different analytical softwares and the laboratory information management system.
Okay.
You are
so the data source
I think you are,
can be manual upload or linked to an existing LMIS.
okay.
I hope that answered your question.
And may I know which all laboratory information management systems you already have shaken hands with or have?
We have Gen 5 softmax.
Sure,
Yes.
thanks.
Thank you.
Jesse, we have one question from Arnab, And he's asking, as you mentioned, you do have a reagent tracking base, but he's asking whether you track, obviously, the critical reagents, the quantity, and the life cycle too.
Yes. So any of our reagents that can be in our inventory will track everything from buffers that have been generated in-house to critical samples or critical reagents. So everything that you would use, consumables, reagents, solutions, etc., will all be tracked the same.
Great. That's really nice. And the next question is from Ketan Waria, and he's asking if the Aegis software can share the bioanalytical data to Vinonlin or SAS for pharmaceutical PK parameter calculations.
Sorry, could you repeat that?
Can the data directly be shared for PK calculations?
Shared
to SAS or Vinonlin software?
So the customized link with the system, we would work with them it's all customizable. So we can work and make a direct link with any system that a client would be requiring.
Great. So we have another question from Karthik Arumugam, and he's asking whether the bioanalytical data assessment can be directly submitted to the regulatory dossier submission. Can this data be directly submitted?
Yes, so the report data can be directly. So all of our reports, our baseline template is the FDA regulations from the 2018 bioanalytical validation. So our basic template, it meets those requirements and everything else can be added on. So this can be directly submitted, yes.
Great. There's another question from Surendra Babu. He's asking whether it is also applicable to the small molecule method validation. Can they also be using it?
For the small molecule?
Yes.
Yes.
Oh, wow.
Again, customizable. So we work with clients and can generate anything that they're looking for with these similar statistics. So if there's a requirement for a small molecule, a component can be generated. We will work with them closely and make sure that all of the regulatory things are met. We can adapt all of the parameters for that.
There's another question. From Patanjali, and he's asking, was any integration tested with Watson LIMS, as most of the CROs have this kind of LIMS?
Yes, so we can use Watson templates as for importation for samples, etc. It's been tested, yes.
That's great. He's asking if you could share the cost for the same, because this really this is filling a huge gap, I guess, it's doing a lot of things. So
yes, well, it is cost effective, as we can, the cost will very much depends on what part you are interested in. And we can work with people for those costs. So the email that I had displayed at the bottom will give you a direct link with our people, so we can give you some pricing.
Okay, that'll be nice.
Thank you, Jessie. And Shefali, we would like to present a certificate to Jessie to felicitate her.
Sure. Yeah, Jessie, thank you for this absolutely beautiful talk. And I think a very need of the hour for most of the CROs. Many have been using it, like Patanjali mentioned, for small molecules, but not many are doing it for large molecules. So that's filling a great gap. And thank you for sharing your software with all of us.
And Shefali would like to add here that a special thanks to Jessie, because I think it is close to 1 a.m. in the morning for her.
Wow.
Close to
Yeah.
10
Yes.
a.m. Really appreciate, Jessie, your time. And I know it would be very selfish on my part to request you to hang on till the final panel discussion. It will be somewhere around 2 a.m. for you. But if you can, then maybe if there are other questions that can be taken up with you in the panel discussion. But if you hang up that should be completely fine.
Yeah.
So thank you, Jessie.
Thank you for having me. It was great.
Srinivas has another question, right?
Oh.
Yes, we have time for one more question, and then maybe we can go to the
It was Patanjali, actually. I was about to ask another question. Do you guys get connectivity with the ETMF documentation through this platform? Electronic ETMF, electronic test master file systems, where all the data goes into that file so that you don't have to generate another aspect of that. Did you guys work with ETMF?
Everything is backed up and archived. So all of the data stays around forever. And it will be in a read-only once it's archived, but it is available for years to come for auditors and reviewers in the future.
Now, the question was connectivity with ETMF, test master files, where the regulatory authorities come back and do the inspection readiness, right? All these documents are deposited there. So you have that connectivity with the ETMF systems.
Yes, yeah. So that's all configurable, yeah.
Okay, thank you.
Thank you. Thank you so much.
Yeah.
Thank you, Jessie.
Yeah, thank you.
So may I now request Srinivas to introduce our second speaker?
Sure, Deepak. Yeah, so it's our pleasure to welcome Dr. Vinay Bathineni, the next speaker, to share his insights onto this theme of technological advancements under the Reg Bio session of APA India 2021. So Dr. Vinay Bathineni holds a PhD from JNTU Hyderabad and has a demonstrated history of working in microbiology, environmental science and technology, and chromatography fields. He has been proficient in R&D management, sales, business development, and implementation of method development solutions in clinical, bioanalytical, and toxicological industries. He's an astute professional and working in highly competitive environment, and of course, meeting the stern goals when you are into a business development role. So he has done incredible work in his master's, which led to his PhD research. And then he has published more than 10 research publications in reputed international journals. And he has been speaking in various conferences about the technology that he has been endorsing. So he started as an application specialist with Phenomenex in 2013, mainly handling the solid phase extraction technique, and taken the grip of the CRO and BAB market for the last 6 years. So with his experience in sales and application support in this competitive market, he's now the business development manager at Phenomenex India, and is responsible for most of the training avenues like seminars, webinars, and even global coordination and market development for all the product lines that actually are supportive to clinical, BAB, and toxicology market segments in India. So welcome, Vinay, and over to you.
Thank you so much for the wonderful introduction. I'm honored, and I hope my voice is clear.
Yes.
Thank you so much. So if you allow me, I'll share the screen.
Please.
So please let me know if you can see the full screen.
Yes.
Yeah, thank you so much. Hello, good morning, good evening, good afternoon, wherever you are from. I'm Vinay, and I welcome you all for today's session. I hope all of you are safe and healthy out there. So without further ado, let me start today's session, that is latest developments in sample prep, how to achieve high sensitivities in simplified liquid extraction and faster solid phase extraction for bioanalytical applications. So before going to the presentation, I'd like to quickly let you know about our external segment reporting structure to the Danaher. We have all these segments: life sciences, diagnostics, dental, environmental, and applied solutions reporting to Danaher. I'm sure you might have heard of these names, some of these names at least, right? So this is the overview of today's presentation. I'll talk about some math, which is important in calculation of our results, and the latest developments in SPE and some case studies, and latest developments in SLE, that is simplified liquid extraction, and some case studies. In any bioanalysis, the widely used term is recovery, right? So the actual tussle will start if we do not get enough recoveries. So this is where we face the actual problem. So what it is, what is it? So it is the calculation used to determine the losses that can occur during the sample preparation process. In simpler way, it will tell you how well is your SP cartridge working. So in assessing the peak area between pre-spike versus post-spike, we can tell how efficient is the extraction. Sample versus reference in a simpler way. And another calculation is matrix factor response. We will compare the post-spike versus neat standard. If the value is greater than 1, it is enhancement, and if it is less than 1, it is suppression. And which we do not want either of these. Ideally, the value should be in around 1, right? So why is it so important? So it is a good indicative to know how good is my sensitivity with the standards versus how good is my sensitivity in the matrix. And then as a result, how good is my sample cleanup? How good am I to achieve an optimal performance of my instrument in simpler way, right? So these two, I think I'll show you some of the examples on these two with the latest developments. As I spoke about the selectivity, so there are so many sample prep techniques, and all of them, all of those techniques will have different selectivities, from basic dilute and shoot all the way up to the SPE as the most selective technique, SPE as in the solid phase extraction. So while stuff at the bottom of the chart is easy to do, but the downside is you may get poor chromatography on the top end of on the top end of this. You can get greater analytical data, but the downside is increased cost, complexity in terms of supplies, energy, manpower, training, all those things, right? So everything has got its own ups and downs. So I'll be talking about the advancements of the top two that are highlighted, that is solid phase extraction, SPE, and supported liquid extraction or simplified liquid extraction, that is SLE. So SPE, as all of you may know, that SPE is the most targeted analyte specific techniques of the entire sample prep portfolio that isolates analyte of interest while removing any interfering compounds that may be present in the sample. In SPE, the goal is to catch and release the target analyte while washing away any kind of interferences. Traditionally, SPE consists of 5 or more steps. One is conditioning. Why am I know most of you are aware of these steps, and why am I stressing is because this is related to the recent advancement. So conditioning of the SPE sorbent is will solvate the chemistries and solvate the sorbent of the dryly packed and tightly packed media, and removes any of the air that is entrapped. So that is the importance of conditioning. And equilibration step, the next one is of the equilibration of the SPE sorbent. Purpose is to remove any kind of the organic solvent from the conditioning step. and prepare the sorbent for loading to promote a good interaction of the analyte with the sorbent, right? So these two are the important. And the next one is loading. You will load your sample, pre-treated sample. And a wash step is to remove any kind of weakly or weakly bounded impurities. And a drying step is also implemented in it can be skipped in some methods, but mostly for good recoveries, drying is important. And the final step is elution of the desired analyte fraction, right? So these are all the steps. So now, while SPE definitely it provides a benefit for analysis, no, it is often perceived as a complicated and time-consuming step, and of course a costly affair too. So so these kind of issues advert some potential users from adopting this technique. So how can we do it more better with this regular SPE? So these are the steps, right? So can we reduce the time spent on SPE? Can we remove conditioning, and can we remove equilibration with just loading your sample with a fixed wash and fixed elution solvent? However, wash can be done, wash and elution are modified case-to-case basis, right? So analyte to analyte. So yeah, can we do this? Yes. Yeah, with the recent advancement, the recent technology is StrataX Pro. Traditionally, you can see on the extreme left side, the traditional SPE methods have required this conditioning equilibration steps to prepare the sorbent for the sample introduction, right? So the conditioning step was again required to wet the sorbent and allow the liquid to enter the pores of the sorbent and enabling the retention within the sorbent, right? So that was it. And the second step, equilibration, why am I reiterating is because we are removing these things. How can we assess, how can we have the good result without these steps? So now once the sorbent is wet, it needs to be equilibrated with aqueous solution. I'm talking about the reverse phase ones, aqueous solution, to prepare it for the sample loading. Now the latest technology, we named it as StrataX Pro, as I said. StrataX Pro so is a water wettable sorbent. The analyte can interact with the sorbent and retained within whenever we load directly onto the cartridge. And in an aqueous solution, we will load into the aqueous solution, right? In reverse phase, mostly we will load into aqueous. And this will eliminate the conditioning and equilibration, because the sample that we are loading, whatever the aqueous that we are loading will wet the sorbent, will condition it and equilibrate it. So these with this new technology, we can eliminate the new steps, conditioning and equilibration. And how does it give? What am I going to get out of this removing? I don't have any issues of conditioning and equilibration. Why are you telling this? So yes, the result is an average reduction of 70% of the solvent consumption and a 40% of the time saving in sample prep time, right? So these are the most valuable resources. I agree. I know all of you will agree with this. Time and our solvent are most costly resources. So yeah, sorry for that. We have two kinds of SP, that rapid and super express methods. Rapid is one thing with 3 steps and super express with 2 steps. So rapid is, I can say, it's a retentive SPE, and super express is non-retentive SPE. I'll talk about this in a while. The 3-step SPE protocol will remove matrix interferences, including salts, any kind of salts, any kind of phospholipids and proteins that are present in the matrix. So this technique will allow for sample concentration and enrichment. So it is the perfect solution for any kind of routine bioanalytical sample cleanup, or you can simply replace with whatever the current reverse phase extractions that you are doing. You can simply do with this actually. So it's a novel polymeric sorbent. And okay, I have some case studies too. in bioanalytical methods, the real tussle is with the polar molecules, right? We may not get the polar molecules recovery as much as with the others. So StrataX Pro is a polar polymer, so it gives a good recovery for more polar compounds. For a panel of acids, neutrals, and bases from plasma, the new technology StrataX Pro provides high recovery, especially for the polar analytes. So analyte recovery is not compromised. Usually sometimes we will have to compromise with the analyte recovery with the wash, right? So analyte recovery is not compromised. And here we can see the better recovery also of for the analytes metoprolol, acetaminophen, and ranitidine, which are the most polar analytes in the suite. So these two, probably the two analytes which are the most polar in the panel are this acetaminophen and ranitidine, right? So during any reverse phase extraction, an increase in the recovery shows that more polar analytes are retained more strongly in this polar sorbent. So, and one more thing is acetaminophen is a weak acid and ranitidine is a moderate base, right? So indicating that this new technology is effective in retaining both polar acids and bases with simple load and fixed dilution solvent. Fixed dilution solvent as in 0.1% formic acid in ACN methanol in 90:10 ratio. So this is about the polar compound recovery. And also I spoke about the matrix factor response, right? So matrix components, especially the phospholipids, will have a negative impact on overall LC analysis and can even degrade system components over time. So our LCMS ion source will be deteriorated over the time because of this phospholipids. So it is important to ensure that these interferences are completely removed. As you all know, choice of wash solvent will definitely influence both analyte recovery and sample cleanliness. Often, the conditions are compromised to allow for either cleaner sample or highest recovery. Sometimes when I speak to the customer, they'll say that, no, I'm good with recovery, but I want the matrix interference should be removed. And some will say that I want recovery and matrix interference, I'm okay with it, right? So, but this will solve the problem. The new technology StrataX Pro, the generic 5% methanol wash, again, the wash will be optimized case to case, right? So we'll provide better matrix factor response for the majority of the analytes in the suite, balancing recovery and cleanup. Yeah, here is another example of increased sensitivity by reducing ion suppression. Here, the 25-hydroxy vitamin D3 is extracted from plasma using StrataX Pro, which has resulted in 3 times more sensitive method compared to the traditional polymeric SPE. And of course, it will save 40% of the method time. And this image shows that LC-MSMS total ion current, TIC, comparison of blank plasma with StrataX Pro, with the protein precipitation compared with the StrataX Pro. Well, why am I talking about these phospholipids again? So, well, I would say these phospholipids, I would consider these are as culprits in bioanalysis that can cause damage to the LC columns as well as ion suppression or enhancement, which ultimately causes inaccurate results. How? The polar head group of this phospholipid can interfere with the mass spec ionization and causing ion suppression. And while the hydrophobic tail can negatively impact reverse phase HPLC analysis because it will strongly interact with the reverse phase column chemistries, leading to a decreased column lifetime. So I have seen, and one more thing, I have seen some of the methods they were using protein precipitation before this solid phase extraction. Some of the methods like metformin combinations, right? So, but this StrataX Pro will remove all the proteins and phospholipids that will be, and that will be ultimately protecting your LCMS and of course column lifetime. One more example. In this application, the sedative hypnotic drugs that are barbiturates, that are phenobarbital, butalbital, and secobarbital. These are extracted from serum using the fast SPE StrataX Pro technology. The serum matrix was cleaned up within the 3 steps, rapid 3-step method, while resulting in high recoveries and low variation between the samples. The standards were spiked at serum at 50 micrograms per ml. Concentration, that was the concentration, and the recoveries were averaging around, you can see, almost 97%. Okay, I can hear some echo. All right. If I may request to go on mute. All right, thank you. And okay. Another one is, though the SP is most selective and targeted analyte specific technique, it can also be used to extract multiple panel of the analytes, right? So while the different panels that I have mentioned here, barbiturates, opiates, analgesics, benzos. So these are all painkillers, sedatives, and tranquilizers, right? So this suite of analytes are highlighted here to show the ease of method development. How you may ask me, how can these variety of these pK, log P values be isolated in a single run? By just maybe, that is why I said optimizing the wash, that is using a stronger percentage of organic. The wash will provide even cleaner results. So we always have to optimize the wash. Though we say fixed wash elution, we always encourage you to do the wash optimization studies. So, okay, in comparison with the traditional protein precipitation method to clean up the serum, the new technology StrataX Pro has shown clear background for multiple panel of the analytes with diverse properties. And the next application is, it is on the peptides, right? So it was done on ion exchange. Usually it is considered that ion exchange will give better cleanup over others. For example, probably it will definitely give cleaner results compared to the reverse phase extraction. But here, the StrataX Pro, the reverse phase extraction shows high recoveries for the extraction of the peptides from plasma when compared to 2 different types of ion exchange polymeric sorbents that are weak cationic exchange and strong anionic exchange. So this method was developed, was improvised, I can say, improvised by adding TFA in the elution step. You can see the TFA in the elution step to increase the recovery of the peptides and show better comparison with these 2 ion exchange sorbent. So weak cation and a strong weak cation and strong anionic exchange. Also, you can see the high recoveries on an average of 77% for all the peptides that are mentioned. Okay, that was about the 3-step protocol, and one last slide on SP I want to show is the 2-step protocol. Other than the catch and release, that is retentive SPE, that 90% of the analysts will do the retentive SPE, right? It can, so along with that, we can also do the non-retentive SPE as a, this can also be used as a chemical filter. So I would say this non-retentive SPE, because we have provided some solutions to the customers with this non-retentive also. The most ignored yet an effective and easy procedure. So non-retentive one. So we can use this 2-step SPE extraction protocol, can remove this matrix interferences quickly. If any of us will have the high concentration of solvents and maybe desalting, some of the methods might need just desalting will be enough. For them, these can be used actually. But the downside is it may not concentrate the analyte as it does in the retentive SPE. Right, so it can be used for multi-residue screening of wet drugs, pesticides, and mycotoxins. Of course, not related to BA, but I just wanted to let you know if you have any desalting, this can be used actually. Oops, okay, non-retentive SPE. And oh, that was about the solid phase extraction. And now let us talk about the another technique, simplified liquid extraction or supported liquid extraction, which is an advanced version of most commonly used technique in bioanalysis, that is liquid extraction. I'm pretty sure most of you will be using LLE too, right? So here are the benefits of LLE and also some of the drawbacks. So when do we do a LLE? So it is definitely useful when you are using wide range of compounds and easy procedure and inexpensive relative to the SPE and rest of the techniques. And there are some drawbacks. So the principle of liquid extraction is the partition coefficient, right? So when we shake it, the two immiscible solvents, because of the formation of the emulsions, the analyte will move from one phase to the other phase. So emulsions, why are these emulsions are drawbacks? Because the emulsions may form causing analyte loss and use of large amount of hazardous solvents and a labor intensive process, and it can be challenging to automate also, right? So that is why we have introduced this SLE, simplified liquid extraction. Supported liquid extraction is a great alternative. What is this? Here is the brief principle about SLE. The liquid extraction with a solid support, a relatively inert scaffolding is used to the liquid extraction here. We named it as Novum. Novum is our specialized for synthetic SLE sorbent. We have others too, right? So the sorbent functions, how does it? The sorbent, the solid support will functions to mimic the shaking step in LLE, liquid extraction, right? So so it is hydrophilic sorbent with it is highly polar. So you can ultimately get a liquid extraction without shaking, no emulsions, and no manual phase separation. I would say even the manual phase separation is the most error-prone technique in bioanalysis, because whenever you are removing one of the phases in LLE, analyte may get breakthrough along with one of the phases coming out, right? So you simply load it, wait for 5 minutes, and elute it with your elution solvent. Hardly we'll have 4 elution solvent, ethyl acetate and DCM, some or the other. So you can imagine a sponge placed on a wet surface, right? So it simply absorbs all the liquid, right? Same way, this SLE will absorb all the liquid. And when you add the elution solvent, the analyte will come off. So one of the major benefits of SLE, that is simplified liquid extraction, is that it is quicker extraction technique than liquid extraction. And it offers approximately 10 minutes of time saving, as you can see. And also omitting the mixing step and no need of centrifugation. Sometimes it might be done, but mostly you can ignore it, because SLE is mostly gravitational pull technique. You don't have to use any centrifuges or any kind of positive pressure or negative. Sometimes it may be used to induce the induce the pull, right? So the manual, and also it will remove the manual phase separation, as I said, right? So no shaking, no emulsion, and no manual phase separation, and it is easily automatable. Okay, probably I would, before going to the two or three applications, I would conclude about this SLE. Probably we can call it as SSLLE, simplified solid supported liquid extraction, right? So it uses a solid support to hold the aqueous samples to prepare for liquid extraction. And you may get the doubt, how can it be? What is the principle behind it? The particle size of the solid support will mimic the shaking step in liquid extraction. In order with that, we can simply eliminate any kind of the shaking emulsion formation, right? So increase in the surface area and reducing the diffusion distance will promote the rapid and efficient extraction. So this is the principle behind this SLE. Passing a solvent through the solid support allows for the extraction to take place and eliminate the necessity of manual phase separation, right? So this is it. And Yeah, we have two types of SLE sorbents, that is synthetic novum, which I was talking about. And also we have another technique, that is diatomaceous earth. However, for bioanalysis, we recommend to go with the novum actually. Right. So yes, now let me introduce you the new technology. We also have a new technology, that is Novum Pro, which I was talking about. It is specially designed for the assays requiring for extreme sensitivities, that is even for parts per trillion level. And it is specially manufactured to reduce noisy baselines and improve matrix factor response. It is a fit to purpose QC testing on API 6500 plus, and to ensure clean baseline with each production batch that we make. The take home point here is a product that has been designed to keep up with the increasing assay requirements in terms of increasing lower limit of detection and lower limit of quantification, and improving mass spec compatibility. Mass spec capabilities too. I have a couple of applications to show in terms of parts per trillion levels on hormones. Here you can see in this study, the estrogen analytes, which are estrone, beta estradiol, and estriol, even E2, E3 respectively. The spiked plasma samples diluted with sodium phosphate buffer, loaded and eluted with loaded onto the SLE plate. Novum Pro we have in the plate format actually. So a plate with hexane and ethyl acetate at 1:3 ratio. The chromatogram that you're seeing here is the representative of standards at 2 nanogram. The column that we have used is kinetics biphenyl. Okay, we are showing lower limit of quantification of estrone, beta estradiol, and estriol in the matrix at 10 pg, 10 pg, and 5 pg respectively. You can see the beautiful symmetrical peaks at very high concentration of standards, even E2, E3. The analytes were separated with excellent recovery and lowest level of quantification. This Novum Pro utilized, while manufacturing, we utilized an optimized manufacturing process to provide any kind of background or less background noise, which will lead to the cleaner extracts. We also have last but one application, testosterone from plasma. Same type of study, ended up with the same type of elution solvent. In 1:3 ratio of this hexane and ethyl acetate, the sodium phosphate buffer as a dilution solvent. Regardless of showing 10 pg per ml of LLOQ, you can see much lower level of quantification of the analytes at a much lower level of hormone concentration, which is, I can say, which clinically significant even for problematic steroids. So before talking about the steroids, I would like to show this slide. The good recoveries and low % CV at a diverse levels of concentration and complete cleanup with the latest technology, that is Novum Pro. Now the last application that I'm showing in this is Novum Pro is on steroids. So steroids can often be difficult to extract from a complex matrix due to their sensitive nature and need for a cleaner background. So this Novum Pro SLE 96 well plate resulted high recoveries, more than 80% on an average, and a low variation between the samples. We have used kinetics biphenyl Korchel LC column here. So that was it. And let me talk about the summary. The latest developments in SPE, that is StrataX Pro, which can significantly reduce your method development time and process time, and high recoveries for polar compounds and a large panel of the compounds, better matrix factor response for polar and non-polar compounds, better sensitivity and better cleanup of phospholipids, all the culprits, phospholipids, salts, and the proteins will be removed without any optimization is needed. And that too with 40% of your time saving, the entire process time and also 70% of your solvent consumption. So let me talk about the next one, that is SLE Pro, the latest advancement. Summary is for assays of extreme sensitivities, even for the parts per trillion levels, a fast method with no background, and I would say much less method development. There is no method development, in fact, but to be honest, we will have to choose the elution solvent based on the how sticky is your analyte, is based on the log P of your analyte, right? So, and it will provide a clean sample ready for LCMS. So that's it. That is about the latest developments. And yeah, that's it. Any questions, you may reach out to me. You can ask me, and if you have any questions, you can reach out to vinay@phenomics.com. And if you have any queries, you can reach out to indiainfo@phenomics.com. And if in any due course, you can reach out to us at this WhatsApp number mentioned, 9 to 6, Monday through Friday. And we have live chat option also 24/7. Globally, we have our technical experts are available throughout, and you can see that at phenomics.com/chat. Right. So thank you. And any questions, please?
Thank you, Vinay, and may I request you to stop the screen share so that your video can be, yes, great. And would request the moderator, Shefali Amarnath Srinivas, to take up the questions, please. And would also request Jessie, if she's around, if you can also be there, then maybe we can continue this with the panel discussion, and few questions, if were not covered for you, even that can be taken up. So Jesse, if you are around, and then over to Amarnath and Shefali. Thank you.
Thank you, Deepak. I think the Srini is not there, but Jesse would also be there in the panel now.
Yes, you can start with Vinay, and meanwhile I'll figure out if Jesse is around.
No problem. So thank you, Vinay, for giving a wonderful talk and giving a lot of insight of the product. Before taking a question from the participant, I would have a couple of questions. So let me ask. And you are based in Hyderabad?
Yes.
Okay. So thank you. So I'm also from Hyderabad.
I'm
working with Beatrice. So there are some commercial questions first. I think some of the participants I also asked this question. So how old this product is? Strata Pro Rapid as well as the Strata Pro Super X. Is it a new technology, or is it the old one?
1.5 years, probably, I can say. And the applications are, we are doing the application part on day-to-day basis based on the need, the hot molecules availability based on the market. But it is 1.5 years to 2 years technology, I can say.
Okay, so all the testing is done and now it's commercially available to supply to the, right?
Yes.
Okay. The another question is in terms of the pricing of these cartridges compared to your, the traditional SP cartridges.
Okay, I would say it will be a little bit, very little bit higher than the regular reverse phase cartridges, but pricing part, I think our team will definitely work on that. That is not a problem, but it will be placed a little bit higher than the reverse phase one based on the advanced technology. I mean, so we will be placing it a little bit higher. That's it.
Okay, and how old the Novum technology is?
It is pretty new, I can say. Novum is kind of a three-year-old, but the Novum Pro is newborn baby, I can say. So we have recently launched this year 2021 for specially targeted for low sensitive hormones, and as you know the challenges of this, we can get it in the extreme levels of even parts per trillion levels. So we are targeting for that for those kind of markets actually. Yeah. So yeah, it is pretty new. This year we have launched, and yeah, it is available in plate format.
Okay, so since you are saying that, the NOM is a new technology, I don't think that you have skills applied to the typical BAB lab.
Yeah, clinical toxicology. Some of the labs will also do on this hormones, right? So these people definitely can opt for this actually. We have tested on this hormones, vitamins, and steroids. So if any bioanalytical lab, they are working on this, they can simply go for this without any kind of method development as that we do for a solid phase extraction.
Okay, so now let me take a couple of questions from the other participants.
Yeah, Amana, just to add up here, so Jessie is not there because it's 2 a.m. for her, so probably she will not be able to join. So I think we'll continue this panel discussion and feel free to take up all the questions for Vinay. Thank you.
Thank Deepak. So one of the questions from Mr. Vijay is, does matrix effect reduction include those from the formulations, for example, polyethylene glycol, which is considered to be the best resource to eliminate the same? Can we have the protocol if already available with Phenomenex?
So PEG, we did not try with this Strata X Pro or something, but again, as it is analyte specific, so probably we can really see the possibility. Yes, why not? We can give it a try and see if this can be used for regulated analysis. But I have provided methods for that particular PEG. Maybe you can reach out to me. I mean, whoever have asked, you can reach out to us separately. We can let you know the method, but not the Strata X Pro. Actually, the other ones we have provided the methods actually.
Okay. Another question is from Mr. Ketan is, the compatibility of the stationary phase of Strata X Pro with the basic pH, or if with a high pH?
Okay, that's a good question, I can say. So the compatibility, all the polymeric media that we manufacture, the Strata brand will be pH stable, 1 to 12 pH stable. That is no problem at all. So absolutely no issues. And with this, even the same case with the latest technology, Strata X Pro, absolutely no issues. These are quite stable than the silica-based ones. Silica-based ones, we will face certain problems with this pH actually, but for this, definitely no, there will not be any issues.
Okay. Is there any specific, the storage condition for these cartridges? I mean, generally, the typical cartridges can be stored at the room temperature.
Yes, same way. So I can say that, we can store in normal as any kind of SP cartridges that we store, we can store it. And without in a dry packed environment, without direct facing of the light or something. So there is no special storage conditions, just normal, just like any other normal reverse phase cartridges. We can store them.
Okay. As a curiosity, I just wanted to know, what is the life cycle for these cartridges is? Sometimes we procure a lakhs of cartridges and keep for several years. So would it be any impact, not specific to the Strata, the X Pro, but any traditional SP cartridges?
So, okay, I got this question in various other platforms also. Probably this is not related to this, but on an overall thing, there is no shelf life or that is mentioned, that will be mentioned in any of the COS, but usually it is not written anywhere. But as we pack in a dry environment and complete sterile environment, there will not be any kind of issues. And probably what I can say is up to 1 year, we can definitely go with this 1 year or so. But it is not any stipulated format that you cannot use this for 1 year. But as long as we store it in a dry and super good environment, there will not be any kind of issues with these cartridges. So I have seen so many customers were using even the years together, they were using the cartridges, even they procured it very long time back actually. So that will not be a case. Unless one point I would like to add is because these are all polymeric based, and unless it is a natural synthetic sorbent, natural sorbent, there might be a chance of any kind of the color change, because the frits might go through the plasma treatment, the high temperature plasma treatment. They might get the color change, but absolutely the media will never, we will never have any kind of problems with the media as such. But for SPE, I have never came across any of such issues. So yeah, this is my.
Okay, another question is, I'm not sure if you have it covered in your slide deck or not. What is the sesame phase used in the X-Pro cartridges? I mean, what are the chemical modification made?
Okay, so that's a good question. So even when the product was launched, I immediately asked the same question to the product management scientists that they launched. So they said it's a proprietary, but I would say it's a normal reverse phase technology. It's a polymeric, a normal polymeric silica based one. But ideally, it's they said the technology is proprietary. So even I do not know what are the chemical modifications that has been done to the sorbent actually. So it's a reverse phase one, I can say.
Okay. The another point is this cartridges have been used for the non-specific binding, especially for the urine assay. Have you done any application?
Yes, that is, again, I think I did not keep those applications on the urine, and yes, we have done, and we have done even on the coffee matrix, various other matrices we have tried, and yes, yeah, maybe we they can reach out to us personally. We can share the application part. So yeah.
Have you done any application on a nitrosamine impurity in a drug product with these cartridges?
Okay,
I mean, this is one of the, the hot area. Every industry is now working with the nitrosamine.
Yeah. So nitrosamines, we did not try with this products, but there are other, I would say, I mean, based on the knowledge that I have on the nitrosamines, it is the much of the sample prep is might not be needed if we can standardize the column itself. I mean, the column protocol with the sturdy column protocol. I would say that I would I mean, the cases that I have seen so far. So, but for StrataX Pro or Novum Pro, we did not try on nitrosamines, but we have some other solutions for nitrosamines with us. Yeah, we can share that also if they can reach out to us.
Okay, what about the batch to batch reproducibility?
Yeah, of course, it is tested. That is why we have confidently launched the product. You know, batch to batch reproducibility. For example, I want to talk about the StrataX Pro. There is no doubt. And coming to the Novum or maybe SLE technique, SLE technology, we have two products. I said, right? We have StrataX. Sorry, I'm getting StrataX, but it's Novum Pro, and we have another product, Diatomaceous Earth, actually. So Diatomaceous Earth, though it's our product, I would directly say that there might be a chance of lot-to-lot variation, because these were taken from the mines directly. So each mine will vary. The case to case, the structure will vary from mine to mine. So unlike the synthetic one, so Novum Pro and StrataX both are synthetic ones, so there will not be any kind of issues in the batch to batch. Yes, we have done the batch to batch studies. Then only we launched the product. So, but for Diatomaceous Earth, that is natural product, we might face some lot-to-lot variations for sure. Not on every case, but sometimes. Yeah.
So Amarnath, this is Pat Patanjali. In that case, how do you kind of bridge those situations? Like when you are in a study which has long number of samples spread over different shipments, and if for some reason one lot is done with, and then they're going to go to the second lot, and that lot doesn't match with the earlier lot, what is the recourse for such a situation then?
Okay, so that is why for bioanalysis, we may not encourage the Diatomaceous Earth product as much as this synthetic one. That is why we place this synthetic one in front, but that doesn't mean that diatomaceous is downside, but it can be used for any kind of the food matrices that will be made in 60 ml capacity. I mean, huge capacities actually. So that is why we will always put this synthetic one. That is safe bet. So we will always promote this for bioanalysis for the low levels of the thing. But for any kind of the analysis that we need to have a huge amount of the samples, then only we will recommend to go with the diatomaceous. But for these SPE and Novum, there will not be any kind of batch to batch issues for sure.
So when a clinical study is there, if a sponsor or if a client comes to you, say we have around 3,000 samples and the cartridges of one single lot needs to be available, you guys make that kind of procurement availability?
Yes, sure.
Manageable?
We can. Yes, I'm sorry. Yeah. Yes, we can do that. We can do that case to case, because recently we have had a discussion on this very recently, and so for one batch, I don't know if I can tell this openly, but yeah, for one batch we can do that. We can do that. That is not a problem. Case to case, if at all we want to have a single batch kind of thing, we can do that. Usually there will be multiple batches, but if it's a special request, we can do that for sure. Yeah.
And the follow-up question on the shelf life for each of these lots, right? So your recommendation is 1 year. Is there any regulatory requirement to specify certain, like 1 year shelf life or anything like that?
That is what I said. When I was telling that 1 year, there is no regulatory guidelines on that. So usually people might consume these cartridges within 1 year, and as long as they are properly packed in a proper environment, so they can stay for as much as so many years also. I have seen, as I said, I have seen many customers were using some of the old cartridges. Some of the boxes, they will be kept on the shelves which they receive as samples, and they will take out and they'll do the tests. Absolutely no problem at all.
Okay.
Yeah.
Thank you, Amarnath.
Thanks, Presently. Vinay, we have one more question from my side. Yeah. What are the different the sizes of this stationary phase? I mean, it can hold with even the 10 microliter plasma to even 1 ml of plasma. What is the different sizes?
Different sizes that we have for you're talking about the SPE or SLE?
SPE. I'm talking about the SPE, and probably you can take this question for your SLE as well.
Okay. So for SPE, we have 30 mg per 1 cc capacity as of now, and we have in plate format too. I wish I had the slide with me, but we have in plate format too. That is 10 megs, 10 mg sorbent in the 96 well plate. So yeah, we have in microelation plate format too, which doesn't need any kind of reconstitution. So for very low levels. So yeah, 2 megs sorbent. The sorbent size is pretty low, 2 megs. So no need of any kind of drying down and reconstitution. You can, after the dilution, we can directly go to the LCMS actually. So that is the thing. So we have in various formats. 1 cc, 30 megs per 1 cc, 10 megs in plate, and 2 megs in microelation plates. Yeah.
The same way for a Novum as well?
Novum, we have it in plate format only as of now, because these are all targeted for very low level low levels of hormones and other steroids, right? So obviously they might not be using tubes as of now, but as of now we have launched in plate format. So tubes we did not launch it yet. So this is a newborn baby, so we are working on it. So probably we should be launching in tube format too very soon.
So did you supply these cartridges in CRO in Hyderabad?
Yes.
And they are regularly using it for typical large amount of the samples?
Oh yes, yeah. StrataX Pro, yes, but not the Novum Pro, to be honest, because Novum Pro we very recently launched this year, and we are promoting. I mean, we are working on the product, especially because we may have to work on this diagnostics. These kind of people might be needing it. But for bioanalysis, that too, as you know, you will agree with me that plate usage is relatively less compared to the tubes usage, right, sir? So
especially in India.
Exactly, especially in India. So we are on that. Actually, we very few of the labs like Clianta, Lambda. I don't know if I can take the names, but no, these people will be, I have seen so far, will be using of plates. Of course, others too, Vida and others too, but the relative to tubes, it is less. Yeah, for Strata X Pro, we have so many are using actually. Pro is especially for metformin combination. It is a huge success actually.
There is a question. There's a question, Amarnath, in the chat. Like, they want to know, there is a lot of clogging in complex matrices happening with these cartridges. So, what is the solution you guys offer for such a situation? I remember a long time ago, several years ago, even for normal SPE cartridges, certain types of matrices, especially even plasma, to having issues with the clogging, and they end up centrifuging those to get the things out. So what is the recourse in that situation, Vinay?
Yeah, thanks for that. Yeah, clogging is definitely a proper sample pre-treatment would definitely solve this issue. Uh, uh, prior to the loading onto the cartridge or plate, we should go with the proper sample pre-treatment. So that is most important. Of course, I did not cover the sample pre-treatment, but again, case-to-case basis.
What is the one of the examples of sample pre-treatment, if you can give that to the audience?
Okay,
what you recommend?
Okay, for example, if it's a whole blood, for let us talk about the whole blood and plasma. For whole blood, we might be needing for urine, you might be needing the hydrolysis sample prior to the loading into the cartridge, whereas for plasma it is relatively less. But for whole blood, we might need to do a proper 3-4 steps prior to the loading into the cartridge. So again, case to case, if it's plasma, we will pre-treat it with maybe one of the things you can observe in any of the reverse phase method. The pre-treatment, whatever the sample, the aqueous one, we will be using definitely water, I mean, during the pre-treatment, and getting it to the pH adjusted to the based on the analyte pKa, we will adjust the pH. So this is one thing, and based on sample to sample, we will, for urine, as I said, we will do hydrolysis, beta glucuronidase, I mean, this hydrolysis. And for plasma, it is relatively easy, but for whole blood, we will do a different kind of sample pre-treatment.
So coming to plasma, so you'll be covering the hemolyzed samples, and other samples are also covered through this pre-treatment itself to address those issues, right?
Yeah.
If the certain hemolysis is happening in the plasma samples, like different grades of hemolysis, different grades of contribution from the lipids, some highly lipid blood plasma samples. So you're going to cover all these aspects to pre-treatment itself.
Yes. So some of the pre-treat, yeah, definitely, I definitely agree with that. So as I said, some of the people will be using prior to SPE, they will be doing protein precipitation for sure, and they will ignore the phospholipids. So these phospholipids will definitely are the culprits for LCMS and the columns. So they might ignore, they would think that I have LCMS, why do I have to spend so much on the sample prep? But the most of the errors that occur are if we do proper sample prep and proper sample pre-treatment, we can avoid these kind of errors. So yeah, so again, you rightly said, you dot on. So phospholipids will definitely deteriorate. I think in a couple of the slides I have reiterating the point about the phospholipids. So yeah, so the sample pre-treatment, this Strata X Pro can in turn will remove these phospholipids. Of course, some pre-treatment should be there, right, case to case, and analyte to analyte. These will remove these phospholipids, proteins, and all the other interfering compounds.
Yeah, thank you for a nice talk. I wish I had this technology 25 years ago when I was working on the bench. So there you go. Advancement to the level, like, I wish certain systems were available for me. Thank you, Amarnath, for giving me the opportunity.
Yeah, thank you. We have the questions regarding, do you have any experience with the tissue analysis with these cartridges?
Yes, of course. Tissues too. That is, I wish I had kept that slide also. How can we do the sample pre-treatment? Because for tissues, I really came across very less compared to the plasma and these things. But yes, for tissue analysis, we did too. We have sample pre-treatment. I think if I can pull out the slide, it would be more helpful, but I'm not sure if I have this readily available. The tissue sample, how to do the tissue sample pre-treatment. Yes, we did actually. So sample pre-treatment, I think we are getting a lot of questions. I wish I had included
Even
this
for
slide
the
actually.
blood samples and all, there is a lot of questions that how the
then
pre-treatment of blood sample tissue can be done. So probably those participants can send a mail to you directly and get it clarified. And if needed, probably your team can reach out to their lab, give some demo, demonstrate them, because this is a new technology.
Yes.
And everyone is live to see that the live demo, because something is there in application and how it works in their lab, probably that would be the real scenario. So probably that the participant can reach out to you over a phone or WhatsApp or mail, and they can invite you to their lab, and then they can get in the real experience with this cartridges.
Oh, we are very happy to do that actually.
So thank you, Vinay, and thank you, Amarnath, and very well said. I think we all always talk about technological advancement in terms of instrumentation. More and more sensitive instruments is what we go for. We look for. But it is equally important to invest similar resources, time, energy, and money into the sample processing as well. So I think it was very well conceived talk, and it was well received with so many questions from participants, and I'm still seeing few questions coming in. So those questions we'll also forward it to Vinay. So thank you, Vinay, for a wonderful talk and a very informative session. Amarnath, you would like to felicitate him by giving him a a certificate.
Yeah, absolutely. Very interesting, the topic, Vinay. And I think, in India, there is a lot of CROs who are day and night, they are working with a lot of, the biochemical samples. And probably, I'm not sure that how many labs are using this cartridge. In fact, we are not using it, but certainly we will invite you in our lab and then, just demonstrate with a couple of the product. We'll see how it works. So,
thank you so much.
On the behalf of even on the APA, we would like to thank Vinay for giving us such a wonderful talk. And I'm sure that the majority of, the participants who are from the typically from the biochemistry lab, they will have a lot of other questions, and that question would be, further send it to you.
Definitely.
And really this technology would help. And the Deepak rightly says that, everybody is looking the sensitivity of the instrument and all, but on the other part, the people should also look at how best they clean up their samples to, enhance their sensitivity, even with the low sensitive, the instrument as well. So thanks for giving us such a wonderful talk, and definitely you may get a call from them, many of the participants, they come and demonstrate your product.
I'm much more happy for that actually. So,
thank you, Vinay, and thank you, Amarnath.
Thank you.
With that, we come to the end of the webinar, and let me just quickly run the poll. So for all the participants, I'm just going to launch this poll. So on your screen, you will see 5 questions about today's talk. Would humbly request all of you to just respond to those questions and submit the response, so that we can then get the list of all the poll responses immediately. And then we go to the next last but not the least activity of this webinar. So I'm going to launch it now, and there it goes live. So on your screen, you should be able to now see a poll, and feel free to just respond to those questions. I will keep it on for a few minutes so that you have sufficient time to respond. I see a lot of responses coming in. Just another 2 minutes, and then we will end the poll. All right, so I think let me just stop the poll. So I'm going to just end this polling. So here we go. And I'm going to download the results. Okay, so I have the results with me now. I mean, results in the sense your responses have been noted. And before I stop this live stream, just wanted to share the talk, the webinar 5 agenda, so that at least before all of you sign off, you should know what webinar what agenda we have for the next week webinar. So hopefully you should be able to see my screen. So next Saturday, 17th of July, we are having a discovery and biotransformation session. The monthly theme remains the same, technological advancements. And we have got, again, yet interesting talks. The first talk would be from June Q, University of New York, recent efforts in ultra-sensitive targeted protein quantification using FAME's LC-SRM MS. Followed by Rini Wink from Nanotemper. He's going to talk about solutions for today's challenging drug discovery pipeline. And then we have GN Albert from SciEx talking about a new era of precision bioanalysis using SciEx 7500 LC-MS. So this is the agenda for the next Saturday webinars. So would request all of you to please join the next week webinar as well. So with that, let me just stop the share here, and I would